Hello, this is Gina Piscatelli with the second lecture for the urinary system in Anatomy and Physiology 2 at Madison Area Technical College. In this lecture, we'll look in more detail at the structure of nephrons, specifically the parts and what functions are performed in each part of the nephron. The pictures that you're looking at on this first slide include a cross-section and a microscopic image on the left of a renal corpuscle, as well as the nephron tubules that are shown in cross-section. And notice most of them are simple cuboidal epithelium. Then the picture on the right shows several glomeruli capillaries, and in some cases you can actually see the afferent arteriole going to it and the efferent arteriole leaving the glomerular capsule, or the glomerular capillaries. So let's first review the parts that we know of a nephron, and this is a diagram of a typical juxtamedullary nephron. I will say that in learning this anatomy, it's probably helpful to draw your own nephron, be able to draw a renal corpuscle, which is shown in the diagram up here, the proximal convoluted tubule, the loop of Henle or Nephron loop, the distal convoluted tubule, and then a collecting duct. And you don't have to be very specific about the winding pathway of the convoluted tubules. One thing to note is the renal corpuscle contains two structures: Bowman's capsule, which is kind of the covering, surrounding the capillaries, which we refer to as the glomerulus or the glomerular capillaries. In addition, notice that there are thicker and thinner segments in each limb of the loop of Henle. We talked previously about how the nephron extending into the medulla and entering the loop is called the descending limb. And then as it travels back up to the cortex, we call this the ascending limb, because that's the way that fluid moves through it. Down through the descending loop or limb, and then up through the ascending limb. But the difference in thickness will become important when we talk about the processes of resorption and secretion. So I'm just going to mention that there is a thick segment in the descending limb and a thin segment, and that will make a difference later in our discussion. So now we're going to look in more detail at the structure of the renal corpuscle. What you know so far is that there is this capsule called Bowman's capsule or the glomerular capsule. There's a capillary network called the glomerular capillaries inside. And you know that blood enters the capillaries in an afferent arteriole and leaves through the efferent arteriole. Okay, the other things that I need to mention is that this is where filtration of blood takes place, and that Bowman's capsule consists of two layers actually. The outermost layer is called the parietal layer, and that's what we generally think of as this capsule here. And it's continuous with the proximal convoluted tubules wall. But there's an inner layer as well, and that's called the visceral layer. And the visceral layer is shown with these kind of funny looking cells that are yellow adhering to the capillaries. Those are called podocytes because they have foot processes. And those podocytes make up the visceral layer. And there's a space in between the parietal layer and the visceral layer called the capsular space. So there's some features of the glomerular capillaries as well as the visceral layer of Bowman's capsule that are ideal for filtration. First is the fact that the glomerular capillaries are fenestrated. Fenestration is a hole, and so notice that there's a pore. There are pores present in the capillary wall. That means that these capillaries are leaky. And then notice that the visceral layer, composed of these podocytes with the foot processes coming out, form these slits in between foot processes or extensions of foot processes. It's called a filtration slit. And the filtration slit will actually limit what can enter the capsular space, which surrounds this capillary. Before we talk about filtration and the membrane for filtration, is what we call it when it has to pass from the blood into the capsular space, let's recall the processes involved in forming urine. So your study question says, recall and state the three main processes involved in urine formation: filtration, reabsorption, and secretion. During filtration, substances are going to move out of the blood, out of the glomerular capillaries, and into this capsular space right here. It will, of course, move into the proximal convoluted tubule as well. So during filtration, the fluid that makes it out of the blood is called filtrate. Then as that filtrate passes through the rest of the nephron, the different regions of the nephron, some substances are going to be reabsorbed. We sometimes call it tubular reabsorption or just reabsorption. And so that means that a substance will move out of the nephron into the interstitial space and then finally into the blood. So it's like recapturing something that's been filtered out of the blood. The last process, tubular secretion or just secretion, is when substances move from the blood to the interstitial space and then into the nephron tubule. So this is a way of getting rid of substances that were in the bloodstream or in the interstitial fluid in a method other than filtration, something that wasn't filtered out but needs to be pumped into the nephron to become part of urine. Now we're going to focus mainly on filtration. And the first topic is, in talking about how filtration works, is to examine the structure of the filtration membrane. What has to be passed or crossed to get into the nephron? And there's three different layers or components of this membrane that blood components have to pass across to get into the nephron. First, of course, they have to get out of the capillary network, the glomerular capillaries, and they use the fenestrations to do that typically. The fenestrations of these glomerular capillaries restricts what gets out of the blood based on its size. So the restriction for getting out of the blood is how big are you? So most things can pass except for large molecules like proteins. So plasma proteins will stay in, and of course blood cells will stay in the blood. They will not become part of the filtrate, unless disease is present, of course. Then there's a layer called the basement membrane, and it's made up of collagen and some other proteins, laminin, nitogen, and heparin sulfate proteoglycan, which is a carbohydrate protein mixture. You can think of it as like any epithelial basement membrane, and it restricts filtration based on size, because to get through this proteinaceous network basement membrane, the bigger you are, the more hindered you are, but also based on charge. This basement membrane repels negatively charged plasma proteins and ions. So they won't make it past the basement membrane. They'll have to go back into the glomerular capillary and out the efferent arteriole. The third component of the filtration membrane are those filtration slits formed by the podocyte foot processes. So recall, this is the capsule's visceral layer. And those filtration slits restrict what can pass based on their size. The filtration slits allow small molecules such as water, glucose, single amino acids, and nitrogenous wastes like urea to pass through. So I have more of a diagrammatic view to talk about these three different components, which might help you make sense of what the filtration membrane is. So the pink is a glomerular capillary. So this is blood, the pink part, and you can see a red blood cell. And then the parietal layer of the capsule is over here on the right. So that's the outermost layer of Bowman's capsule. And here we have the capsular space where the filtrate will be. This is where filtrate will accumulate initially. I'm running out of room. Sorry, it's so slow. And then the visceral layer forms those filtration slits. So that's part of the filtration membrane. And in between the glomerular capillary wall and the visceral layer is a basement membrane. So substances that are going to become part of the filtrate in the capsular space have to first get through the fenestrations of the capillary wall, cross the basement membrane, which is a meshwork of proteins and carbohydrates, and then make it through the filtration slits of the visceral layer of the capsule. Now those filtration slits have a network of molecules that form a diaphragm, so that really limits the size down to like 3 nanometers, what can fit through there. The amount of filtrate that makes it across those filtration membranes in both kidneys, all the nephrons, all the filtration membranes, is about 120 to 125 milliliters per minute. That's a lot. That equals 180 liters per day. But we only urinate approximately 1 to 2 liters per day. Of course, that depends on your hydration status, what you eat or drink, any drugs involved, etc. But regardless, it won't vary that much. Most of the time, 99% of what has been filtered out of the blood has to be reabsorbed from the nephron back into the capillaries. Whatever that 1% of filtrate that's left is going to be the urine. So the filtrate has to be converted into urine. And the processes involved with that are reabsorption and secretion, which we've already talked about. Now, we call the fluid that escapes the blood or is filtered out of the blood filtrate. And then as it moves, this fluid or filtrate moves through the nephron, its composition is going to change. So we just call it tubular fluid until it gets into the renal pelvis, and then we can call it urine because then it's in its final form. So the processes of reabsorption, something is reabsorbed out, or secretion, that is when something from the surrounding tissue gets put into the nephron, will happen in different locations of this nephron and will depend on the wall of the nephron's permeability. So the permeability is selective in different regions of the nephron. And it will also depend on mechanisms of membrane transport that occur in the proximal convoluted tubule, the loop, or the distal convoluted tubule, as well as the collecting duct. As an overview, we can say that 65% of the filtrate is reabsorbed in the proximal convoluted tubule, and 25% is reabsorbed in the loop of Henle. That leaves just 10% or 9% to be reabsorbed in the collecting duct there, and then the rest will become urine. Now, so reabsorption happens in 3 major locations: proximal convoluted tubule, loop of Henle, and the collecting duct. Secretion, by contrast, happens in the distal convoluted tubule, where it will largely be based on hormones, as is reabsorption in that location, but there'll be some secretion there, and most secretion will happen in the proximal convoluted tubule. So I think it's fair to say the proximal convoluted tubule is the most important site for both reabsorption and secretion. One last anatomical topic that we have to discuss is related to the way we've been drawing nephrons and the fact that renal corpuscles aren't really oriented the way we've been showing you. They're oriented in such a way that there is a physical connection between the end of the nephron loop and both the afferent and efferent arterioles. So we've been drawing the nephron such that you can easily make out the beginning renal corpuscle, the proximal convoluted tubule, da-da-da-da-da-da-da-da-da-da. In actuality, these arterioles make contact with this ascending limb of the loop of Henle just before the distal convoluted tubule. So what that means is you kind of have to flip this whole renal corpuscle and proximal convoluted tubule 180 degrees. So I've done that on this picture on the right. So now we have the renal corpuscle facing this end of the loop and the beginning of that distal convoluted tubule. And there is a physical connection between these arterioles and the wall of that ascending limb of the loop of Henle. And it has a name, this region, this connection, and it's called the juxtaglomerular complex or juxtaglomerular apparatus. Remember, juxta means near. Glomerular just refers to the glomerular capillaries. And so the physical connection that occurs right here is called the juxtaglomerular complex, or apparatus. I know it as apparatus, so I often call it the JGA, but it could also be called the JGC. JGC, yeah. Anyway, let's look at a picture of it instead of my attempt at drawing. So the nephron on the left is really the way we should have been teaching it to you. Here is the renal corpuscle and the proximal convoluted tubule, descending limb, ascending limb of the loop, and now the distal convoluted tubule begins. The collecting duct isn't even on here. So if we look in more detail, more magnified view, the region that the box is showing here, we see that there is a connection, these purple cells right here are part of the ascending limb of the loop of Henle, some extra cells, as well as the wall of these arterioles. So there's some physical connections here. And the reason this complex is important is because all the different cell types that are located in it regulate the rate at which the blood is filtered. So we call this glomerular filtration, and it happens at a certain speed based on blood flow, and it can be slowed down or sped up based on the interactions of these cells. So we just want to first understand what cells are here, and then in the next lecture we'll talk about what they do. So the components of the juxtaglomerular complex include three types of cells. The walls of the afferent arteriole in particular are thickened, and there's smooth muscle cells there. They're called granular cells. Sometimes they're called JG cells, juxtaglomerular cells. But anyway, they are the walls of the afferent arteriole. Now these cells, as you can imagine, since they're smooth muscle, can contract, and that would constrict the afferent arteriole. But these cells are so unique, they also secrete a hormone that you've heard of called renin. So these are very unique smooth muscle cells. Not only can they constrict and contract and cause vasoconstriction, but they make a hormone, renin. And that renin, if you remember, is part of the angiotensin II pathway, and so controls blood pressure. In addition, these smooth muscle cells act as mechanoreceptors to detect the blood pressure within this afferent arteriole. And we'll talk about why that's important a little bit later. The second component of the juxtaglomerular complex is called the macula densa. And these cells include part of the ascending limb cells that make up that wall. We'll just leave it there. That's what it includes, macula densa. And these cells are chemoreceptors. So tubular fluid that is ascending up the loop of Henle has a certain concentration of sodium chloride. And the macula densa cells can detect whether or not that sodium chloride content is too high or too low. Now, what is going to happen in response to that will probably be some sort of resorption or secretion, but we'll talk about that later. The third component of the juxtaglomerular complex are the mesangial cells. And they're kind of in between the other two types. They form this triangle here. Okay. And so the mesangial cells are cells that do signaling because they have gap junctions in between them and signals can pass easily. But they pass signals between the macula densa cells and the granular cells. So 3 components. And with this introduction, we're going to be able next time to focus specifically on filtration and how it's controlled and regulated. Thank you. That's the end of this lecture.